That is the histone core. And in the histone core, each unit, that is the H2A, H2B, H3 and H4 are present in dimeric form. किस फॉर्म में है? डायमेरिक में दो का है. And in this way it forms the octamer. It forms the octamer. ये क्या बना रहा है? एक octamer को बना रहा है. ठीक है? And after the formation of the octamer, the DNA is wrapped. DNA उस पर क्या कर रहा है? Coil कर रहा है, wrapped कर रहा है. Incomplete 2, that is the 1.9. 1.9 turn. ये कितना बना रहा है? 1.9 turn. And the loops are attached by the histone 1, H1. Clear? कोई दिक्कत नहीं? Now you see here. This one is the DNA. DNA associated with the nucleosome core. Nucleosome core. That is the octamer, and each histone is present in the dimeric form. Clear? Now it is wrapped. Con? DNA. And DNA is wrapped to form the nucleosome. Then it comes the looping. ठीक है? And it forms the 8 structures, 8 nucleosomes, chromatosomes, and it forms the chromatosomes, and it forms the solenoid. It forms the solenoid. Solenoid is a fibrous structure, and it represents the 30 nm fiber. 30 nm fiber. With an axis, एक axis भी यहाँ पर, with an axis, it forms the loops. It forms the loops, and then SK folds around some non-histone proteins, and form the chromatin. Again, it condensed and form the chromosome. It is the organization of eukaryotic chromosome. It is the organization of eukaryotic chromosomes. Here, you observe the condensation. Here you observe the condensation. That is the condensine and cohesion. Condensine and cohesion. These two components are associated here. See here, condensine and cohesin, two proteins. Both are the proteins. The condensine is associated with the specific condensine sites and determine the condensine domain border. मतलब कहाँ से कहाँ तक उसको जुड़ना है, उसको क्या करेगा वो? Border को determine कर देगा. ठीक है? And after the determination, the condensine domain border is determined by the cohesin, and in between the cohesin, the condensine sites are determined by the condensine protein. The cohesin protein determines the limit. The condensine protein is associated, responsible for the condensation. Then they represent the folding. This one attached here and this one attached here, like this, represented by the arrow. Clear? They are folded. In this way they are folded by the activity of the condensine and cohesin, and it forms in this way, you see here, that is the cohesin and this part is the condensine. It forms the loop, it forms the domain. Then what is the difference between the loop and domain? Most important part is here. What is the difference between the loop and domain? Domain determines a major areas मेजर स्पेशियल एरिया, स्पेस में जैसे हम आपको देख रहे थे, मान लीजिए यहाँ पे खड़ा है, ठीक है, हम अगर यहाँ पे खड़ा है तो मेरा एक स्पेस में एरिया ले रहा है ना, ये डोमेन है उसके लिए। किसके लिए? बायोकेमिकल मॉलिक्यूल्स के लिए। बायोकेमिकल मॉलिक्यूल के लिए क्या है? वो डोमेन है। तो दैट रिप्रेजेंट्स ए डोमेन, ठीक है, एंड दैट रिप्रेजेंट्स द लूप। दैट मीन्स इन सिंपल वे you should consider that the number of loops are associated all together to form a domain. Number of loops are associated all together to form a domain. And in this way, a number of domains are formed during the formation of chromatid. During the formation of chromatid. Clear? यहाँ पे कोई दिक्कत नहीं है. Now you see here. The other factors which are responsible for the condensation are different proteinous component described. Different proteinous component described. A proposed model for the sister chromatid cohesion by RSF factor. RS factor 1. RS factor 1. And then RS factor 1 here is responsible for the tip 60. Tip 60. It is also the other protein, tip 60, and it is associated with the K2. मतलब K2 ये मतलब histone 2. Histone 2. Second histone. 2A. Histone कहाँ है? 2A. And the site is the K11-8AC. The site is the K11-8AC. It is the site where it is associated. At chromosome arms, an RSF1 bound SDAC. It is also the factor. It is the factor SDAC at centromere contracts. And after the association, अभी हमने बोला कि यहाँ पे क्या होगा? ये कॉडेसिन जो होगा, इसको आप देखिए यहाँ पर थोड़ा सा आपको ये चीज़ को समझिए, यहाँ पे समझिए इसको. अब यहाँ पे देखिए ये दो पार्ट है, कोहेसिन है और कंडेंसिन है. कंडेंसिन एंड कोहेसिन इज रिस्पॉन्सिबल फॉर द मतलब कोहेसिन इज रिस्पॉन्सिबल फॉर द फोल्डिंग ऑफ द मेजर एरिया इन विच द कंडेंसिन इज प्रेजेंट. They are the loops. The condensin are responsible for the formation of loop, and cohesin are responsible for the formation of the domain. वहाँ पे वो domain बना रहा है. And if you observe the molecular nature, that the activity of the condensin and cohesin are determined by the different factors here. The study of the condensin, because from which site the DNA is folded. क्योंकि fold कौन कर रहा है? DNA. डीएनए किससे एसोसिएट हो चुका है? डीएनए इज एसोसिएटेड विद द हिस्टोन. वो टाइटली बाइंड हो चुका है किससे? हिस्टोन से. दैट मीन्स द टू चीज. मान लीजिए सपोज करके आप एक रस्सी बनाते हैं ना भाई, घर में रस्सी बनाते हैं. अगर आप ये रस्सी बना रहे हैं तो कहाँ कहाँ से आपको मुड़ मुड़ के गोल गोल बनाना है, तो पहले तो उसको आप डिटरमाइन करिएगा कि यहाँ से हमको बनाना है, बांधना है, गांठ देना है, तो वहाँ से लाके गांठ दे दीजिएगा, फिर दूसरा गांठ दे दीजिएगा, और फिर सब गांठ को एक जगह करना है. ठीक है? सेम एक्शन इज ऑकर्ड हियर. एकदम उसको सिंपल वे में समझिए कि एक रस्सी है लंबा सा, रस्सी में पहले छोटा छोटा लूप बनाना है, ऐसे 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 लूप बनाना है, और जब 4 5 6 लूप हो गया तो उसको क्या करना है? सब लूप को एक जगह लाके एक डोमेन बना देना है. ठीक है? तो at first the site from where condensin and cohesin want to fold the strand is necessary. उसको recognition चाहिए, and that is condensin is recognized the site by the SMC2 and SMC4 factors. SMC2 and SMC4, and that on the strand, it recognizes the CAP-D2-D3, that is the condensin site, and CAP-G/G2, that is the condensin site 1 and 3, and that is responsible for the folding. CAP-H/H2 site, that is the condensin site 2, and they are all are responsible for the activity of the condensation. And here the cohesion is also SMC1 and SMC3. That means the SMC1 and SMC3 are associated with the cohesion, and SMC2 and SMC4 are associated with the condensation. याद रखना है कि SMC1 and SMC3 are associated with the cohesion, and SMC2 and SMC4 are associated with the condensation. And then they are, after the recognition, ठीक है, after the recognition of the site, if you have मतलब अगर ये चीज़ आपको याद नहीं रह पाता है तो आप डायरेक्ट भी लिख सकते हैं, after the recognition of the sites they are responsible for the condensation and cohesion of the strands to form the specific domain. And after the formation of the domain, the chromatid is formed, and after the formation of the chromatid they are further condenses to form the two chromatid a chromosome having the two chromatids. Clear? कोई दिक्कत नहीं. And this represents the folding. Here the 3D genomic organization. Here ये जो represent कर रहा है ये 3D को represent कर रहा है, इसीलिए ऐसा है. Condensing recruiting factors. These are the factors. These are the responsible for the condensing. That is the factor. One factor is here, one factor is here. These are the two factors. Okay? And these are responsible for the condensation. One condensing and another condensing. And these are the cohesion. Clear? It is some complex, इसलिए इसको आप छोड़ दीजिए. इसको समझ गए? इसमें कोई दिक्कत नहीं है. Now here